Fetal pacemaker ready for human trials. Researchers at the University of Southern California first developed a micro pacemaker for fetuses 5 years ago, and the device is now ready for its first human trial. The fetal pacemaker is a slim cylinder with components that include a single transistor relaxation oscillator, an epoxy capsule, and a small lithium battery. The pacemaker is implanted into a fetus through a 3.8mm diameter insertion cannula. The battery is able to power the device for about a week. When the power runs low, a high-powered field generator can be used to generate a radio frequency magnetic field outside the body. This wirelessly recharges the battery through inductive coupling. The device, which has been successfully tested in sheep fetuses in the past, was granted humanitarian use in 2015 by the FDA. Say no to safe spaces on YouTube. Support Tomo News at patreon.com/Tomo News. It's amazing how far medical tech has come.
Man's paralyzed limb reanimated with the help of a brain chip. A team from Ohio has made a medical breakthrough, successfully developing technology that allows brain signals to bypass a spinal injury and transmit straight to the muscles. When Ian Burkhart broke his neck 4 years ago, it damaged his spinal cord and left him paralyzed from the chest down. He retained some movement in his shoulders and biceps, but lost sensation in his hands and legs. To help him, doctors inserted a chip the size of an eraser head into his motor cortex, the area of the brain that controls hand movements. The chip records brain signals for specific hand movements and sends these to a computer via a port on the back of Burkhart's head. Once the signals are decoded, they're transmitted to an arm sleeve studded with electrodes. The electrodes stimulate the muscles and allow them to move. The system, called NeuroLife, has allowed Burkhart to make 6 different hand and wrist motions. It marks the first time a paralyzed man has been able to regain movement using recorded brain signals.
Gene-edited immune cells clear babies' incurable leukemia. A baby girl in Britain suffering from leukemia has become the first person in the world to receive an experimental gene-editing procedure that miraculously reversed her cancer. Layla Richards was diagnosed with acute lymphoblastic leukemia when she was just 14 weeks old, a disease in which the bone marrow makes too many immature lymphocytes. This is the most common type of cancer in children. In a healthy child, the bone marrow makes blood stem cells that can become myeloid stem cells, or lymphoid stem cells, which then develop into mature red blood cells, platelets, and white blood cells. However, in a child with acute lymphoblastic leukemia, too many stem cells develop into lymphoblasts, B lymphocytes, or T lymphocytes, which are in fact leukemia cells. Leukemia cells are not able to fight infection, and they take up the space for healthy blood cells in the blood and bone marrow. This may lead to infection, anemia, and easy bleeding. Doctors in London performed a new gene editing technology known as TALEN on Layla, which had previously only been tested on mice. The technology uses molecular tools that act like scissors to cut specific genes in order to make T cells from healthy donors behave in 2 specific ways. First, the cells are able to become invisible to a powerful leukemia drug that would normally kill them. Second, they are reprogrammed to target and fight against leukemia cells only. Layla spent several months in isolation due to her extremely weak immune system after the procedure. After the leukemia cells were confirmed to have been eliminated from Layla's body, she was given a bone marrow transplant to replace her entire blood and immune system. The treatment was prepared by scientists at London's Great Ormond Street Hospital, University College London, and French biotech company Cellectis. Cellectis is going to fund full clinical trials of the therapy starting next year.
Scientists find antibody that makes cancer cells kill each other. A team at the Scripps Research Institute has discovered a new therapy that could prove to be a safer way to battle cancer. Acute myeloid leukemia, AML, is caused by an overproduction of immature white blood cells that are unable to function properly or fight infection, and interfere with normal blood cell production. Current cancer treatments stop the growth of cancer cells but almost always damage other healthy cells. Scientists found that applying certain antibodies to AML cells triggered them to mature into natural killer cells that support the immune system. These natural killer cells then destroy related AML cancer cells, but leave unrelated cancer cells alone, prompting researchers to call the technique fratricidin therapy. In a single day, natural killer cells in one sample destroyed 15% of their cancerous kin, suggesting that a safer, more effective form of treatment could be well on the way. The Scripps team is working to be able to test the new therapy on human patients next year.
Say no to safe spaces on YouTube. Support Tomo News at patreon.com/Tomo News.
Paralyzed man walks again after receiving cell transplant. A paralyzed 38-year-old man has regained the ability to walk after undergoing a breakthrough procedure that repaired his injured spinal cord. The pioneering spinal cord repair procedure was performed in Poland, in collaboration with researchers in London, on a patient paralyzed from the waist down following a knife attack in 2010. For the procedure, olfactory and sheathing cells were taken from one of the patient's olfactory bulbs in his nose, along with strips of nerve tissue from his ankle. The cells, known as OECs, were cultured and injected around the damaged site on the spinal cord. The nerve tissue is grafted across the gap on the spinal cord. The researchers believe that the injected cells stimulated the inert spinal cord cells to regenerate, using the grafted nerve strips as a bridge to fill the gap in the spinal cord. The patient can now walk with a frame. With sufficient funding, 3 more paralyzed patients are expected to be treated in Poland within the next 3 to 5 years.